I was thinking about how to set up this talk today. It's really a neat situation. We don't often have the chance to present our work to a more general audience as well as a scientific specialty audience. So I try to cover all bases here. So I do apologize that I imagine there'll be parts that are simplistic and boring to some people and maybe more complex to others. So the only thing that I have ever wanted to do is be a virologist, which is an oddly specific career choice, and I will take you through that. This will be part of the basic stuff. But I was thinking about how that changes from nowadays to when I was a kid in the 70s and 80s. If I was to ask my son, who is in the back and 10 years old, what he wanted to do as a career, I'm sure he would say a gamer or a YouTuber, which is quite different to what my parents had to deal with a kid who wanted to be a virologist. So in the 80s, and I'm saving you from a video here that I think was posted, but it was a video that scared me a lot. In the 80s, in Britain, which is where I grew up, there was very much a fair campaign about a viral disease, a zoonotic viral disease called rabies. I think most people have probably heard of it, but it is a tremendously dramatic disease that's caused by rabies. Britain, obviously an island, and one of the few places, but not uniquely, but few places in the world that doesn't have rabies. So in the 80s, as I said, there was a fair campaign, a public information campaign, that sought to scare people into not importing animals from continental Europe into Britain. And there is no reason why I would know the French, German, and Spanish for rabies if it wasn't for this public information campaign. I have shivers get down my spine now looking at this. But these sorts of signs were at all of the ports with very dramatic, scary writing telling us not to bring cats and dogs into Britain. And they weren't subtle in the message either, don't smuggle death. And what I was going to show was a public information film, but as I was looking for information for these slides, I was reminded of this movie that, for whatever reason, my parents allowed me to watch in the early 80s, called The Mad Death, which was fabulously scary and portrayed life in Britain if rabies was to become endemic. These had huge influences on me as a 10-year-old. Immensely scary, and again, strangely, I bought this book recently. So I read this book by Colin Kaplan, who was chair of the second oldest microbiology department in the world, which is in the UK. And even though it was very much a book about facts, non-fiction book, it sort of took you through rabies and what it meant. What was remarkable to me at the time, and in truth it still is, that rabies is a virus that manipulates the behavior of the infected individual. I mean, think about that. So it changes how you behave, as well as just transmitting, changes how you behave. It is, for all intents and purposes, 100% lethal. That was also exceptionally scary to me as a kid. But what was equally remarkable, maybe more remarkable, was for more than 100 years now, we've had vaccines for rabies that really lower the case fatality rate down to, in the Western world, approaching zero. That idea that you could protect against something that was so lethal, so dangerous, had a great impression on me. So then, I didn't really understand what viruses were, but I did have a good sense that they were small. So if you look right to left, size of a human hair is up to 200 micrometers. Eukaryotic cell, so a human cell, 10 to 100 micrometers. Yeast, 3 to 10 micrometers. Bacteria, 1 to 10. Viruses are far smaller than that, 0.05 to 0.1 micrometers. So something so tiny can do so much damage. And to put that into context, you can fit more than 3,000 virus particles across the width of a human hair. We can't see them. We need special tools to see them, which is essentially an indirect way of seeing them, and that's electron microscopy. But they can and are exceptionally dangerous. So I mentioned the case fatality rate. What that means is the total number of deaths over the total number of cases times 100. So if we think about viruses that we've probably all heard of that have very high case fatality rates, bird flu, which is in vogue again now, may have up to a 60% case fatality rate. I spent a fair amount of my career working on Ebola virus. Every grant I wrote prior to 2014 described Ebola as having a case fatality rate of 90%. The reality is that it's closer to 50%, but that's nothing to be sniffed at. 50% of those people who are infected die, regardless of the intervention that's offered. And we do have some vaccines that are coming online that may reduce that number. We all know about SARS-CoV-2. That's the agent that caused the COVID-19 pandemic. Case fatality rate of 3 to 4%. All of us may know people who died. It's so recent with such large numbers of people affected. And rabies, really the anomaly here, the most pathogenic virus we know is down to less than 1% of those infected because of the power of vaccines. There have been many examples of pandemics such as we went through with SARS-CoV-2 and COVID-19. This picks out a number of bacterial and viral pandemics, but just to highlight the numbers of individuals that can be affected, I want to highlight the Spanish flu pandemic in the late 19-teens, killed maybe as many as 100 million people. That's an amazing number, especially when the population of the world was a lot smaller than it is now. HIV and AIDS, in certain parts of the world, it might be considered a chronic disease, manageable disease now, but it's certainly been responsible for more than 30 million deaths. And COVID-19, I think there's a fair amount of debate with the numbers, but if we stick with this figure, it's 27 million deaths. So what's the next one? And that's really the area of our research, trying to make sure that we can react quickly to whatever the next pandemic or outbreak would happen to be. So I was given this slide, but it highlights emerging and re-emerging infectious diseases since the 80s. And it doesn't go through to 2025, but since the 80s. And there are some viruses on here and bacteria on here that we'll all know about. So Ebola virus tends to emerge in the middle part of Africa. Mpox, there's currently more than an outbreak now in several parts of the world. Hendra virus and Nipah virus are Asian and Australasian viruses, also with high case fatality rates. I'm not going to walk around this map. But it's not just Asia and Africa that are affected. There is a virus on here from Missouri. And I did not manipulate this at all, but Heartland virus here. Not many people have been infected, but it certainly has a high case fatality rate as far as we can tell. So we have to be watchful and do whatever we can to mitigate the impact of these viruses. So this is very much the focus of the work in our lab. I want to tell you a little bit now about how I got to this point. So I mentioned that I was interested in rabies, which is a neurotropic virus. It's a virus that targets the central nervous system. I grew up in the middle part of the United Kingdom. The part of the country that I really grew up in is not quite as pretty as this, but this is Stratford-upon-Avon, and as far as I can tell, there are more Americans in Stratford-upon-Avon at any one moment than British people. Then for graduate school, I went to the University of Cambridge, as Dave said. This is Queen's College and Mathematical Bridge. During graduate school, I worked on a neurotropic virus. I also did as an undergraduate as well. There was no rabies work at that time in the UK. And my focus for my research during my PhD was herpes simplex virus. So herpes viruses are viruses that hide out in the central nervous system. They're also remarkable in as much as once one is infected for life. So that's an interesting strategy for a virus. I continued working on herpes simplex virus when I went to do my postdoc in Boston. And as Dave said, moved down to San Antonio. And it was in San Antonio that I was given the opportunity to work with essentially anything that I wanted to work with. So as long as we could get funding, we had the facilities that would allow us to work on anything. And it was at this point we started to get interested in emerging infectious diseases, and filoviruses and Ebola virus were a big part of that. Spent more than a decade in San Antonio, moved back to Boston, that's number 5, where I spent 6 years almost at Boston University working in a lab there. And then middle of last year, I moved to Missouri. So it's been moving around, but the evolution of the research has been solely neurotropic viruses through to not really any virus in particular. We really are a mile wide and an inch deep, but our focus is emerging infectious diseases. So these viruses, as I hope I've convinced you, are dangerous. We don't want to put ourselves in any more danger than we need to be, so we need to make sure that we can work with these pathogens in a safe environment. And that means safety for the person doing the work, the laboratorian, also for the environment and the public as well, and other things that aren't listed here. So to do this safely, we rely on a number of different things. One, viruses tend not to be very stable. But importantly, we go back to viruses being small. Many of the safety procedures that we use rely on viruses being small and moved along with air. So we can manipulate air to move from one place to another, and along with that air will move the virus. And it is exploiting the size. So I was sort of hoping that I'd come up with a joke about this, but this was an AI-generated image of a person on a plane with the air vent blowing over their face. I sort of imagine, because this is what I do, that microbiologists are the ones on the plane that definitely have this curtain of air in front of their face to protect them from whatever is floating around. So in this country, and it's similar all over the world with slightly different names, we grade the laboratory environment that we work in for safety reasons on 4 different levels. So in biosafety level 1, and I will describe what this means in a minute, in biosafety level 1, we can work with viruses that don't cause disease in healthy human adults. So certainly when I was in high school, probably, hopefully, what we were doing was biosafety level 1. Biosafety level 2 gets a bit more exotic, so probably most of the university labs, but most of the molecular biology labs out of necessity are also biosafety level 2. So this is viruses or pathogens in general that pose a moderate risk to health, but tends not to be impactful in healthy human adults. And that anomaly, again, the most pathogenic virus we know, because of the mitigation strategies we have, rabies virus, we can work on at biosafety level 2. I keep rambling on about rabies virus. I have not worked with rabies virus. Before I retire, I will work with rabies virus. Herpes simplex virus requires BSL-2 as well. So biosafety level 3, and Dave mentioned that I work at LIDR. This is the containment level we have at LIDR. This is where it starts to get a bit more exotic and a bit more exciting. These are viruses that can cause serious, potentially lethal disease, and we need engineering and protective equipment controls accordingly. Examples of pathogens that need biosafety level 3 containment, West Nile virus, Eastern equine encephalitis virus, Heartland virus from Missouri. Remember, the outside world is biosafety level 4. Biosafety level 4, This describes some of the labs that we had at BU and also when I was in Texas in San Antonio. These are viruses that pose a high risk of lethal disease and that tends not to be a vaccine or a therapy. So, Marburg virus, 50% case fatality rate, Nipah in the same region. No vaccines or therapeutics have been licensed for either of those pathogens. So when we work in biocontainment, we think about the equipment that we wear. So in BSL-2, it's probably exactly as you imagine a scientist to work. This is just an example. Lab coat, let's pretend she's wearing safety goggles, gloves, working in a biosafety cabinet that I'll talk about in a bit. And it doesn't take much in the way of training to work at BSL-2. BSL-3 takes a bit longer to train. You can't just walk in and start handling these viruses and bacteria from the get-go. We'll train people in and make sure that they're working safely. The personal protective equipment that one wears at BSL-3 is somewhat variable, but this is an example of something that's fairly common. Person's wearing a Tyvek suit, has a respirator that's pumping filtered air to this headpiece, and the headpiece has air that's under positive pressure relative to the rest of the room, so it's blowing air away. So that respirator is offering protection from these pathogens that might be floating around. So BSL-4, and it's been a while since I really worked in the lab, but I never had a suit that fitted me as well as this one. And it was my first suit. So this is the sort of garb we wear typically at BSL-4. So it's a fully encapsulating suit that has limited penetrations. It works at positive pressure. So air is fed into the suit. That air serves as breathing air, but it's also, if there happened to be a hole in the suit, it's blowing away any pathogens and keeping the person safe. Beyond that, depending on the pathogen that you're working with, you might need a shower when you leave the lab. So you enter a gray area that's neither clean nor dirty, and you take a personal shower. At BSL-4, you take two showers. You take a shower in the suit. So the first shower will have a disinfectant, and then it will be rinsed off. And then you'll have a personal shower after that. So there's no quick exit from the lab. Obviously, beyond PPE, we have engineering controls. Again, we're relying on the movement of air. So we have negative pressure rooms. Part of the reason it's so complicated to work in containment is getting the air to move in the correct direction is not easy. We're moving around, we're opening the rooms, we're closing the doors. It's not a trivial exercise at all. But we have supply and exhaust air synchronized such that there's more exhaust than supply, and we keep the rooms in a negative pressure state. To make it more complicated, we also want to remove anything that might be floating around in that environment. So we have a lot of air changes in that space, much more than in this room, for example. We also filter the exhaust air. So everything that comes out of the labs we consider might be contaminated. So we pass it through multiple filters to clean that air. We work in these biosafety cabinets. So even with all this engineering, building control to make sure that we're safe, we still, when we're handling viruses or bacteria, we're working in a biosafety cabinet. That keeps us extra safe. Solid waste has to be cooked in an autoclave. That's really an enormous pressure cooker. So it kills everything that goes into that pressure cooker, and we check it. We make sure that every run has killed all of the samples that are in there. Even the liquid waste, we don't just pour it down the sink. We will decontaminate it, maybe soak it in bleach for a period, or it goes into what we call an effluent decontamination system, which is really, again, an enormous pressure cooker that cooks hundreds, sometimes thousands of gallons of liquid at 250 degrees C for 2 hours. So this is the sort of arena that we're working in. It's biocontainment, it's very specialized, it is very expensive, and there aren't many of these laboratories deposited around the country, not even in the world. So this is not a complete map of the US biocontainment capability, but it is a complete map of a subset of the biocontainment facilities in this country. So, I don't know, in the mid-2000s, the US government decided that it needed to make sure that there was capability on university campuses to blend academic expertise with biocontainment. And that includes training the next generation of individuals who work in this environment. So they set up 2 national laboratories and 12 regional laboratories. And the 2 national laboratories work at biosafety level 4. That's the one at BU and the other one at University of Texas Medical Branch in Galveston, Texas. And beyond that, there are the 12 regional labs, of which the University of Missouri won the grant and subsequently built the lab that is now known as LIDR. So this is LIDR. I can't say that a great deal of expense was spared on the design of the building in the way that we have for NextGen. Maybe Dave can design the next one. But we have significant containment capacity in this facility. We can work at BSL-3, as I've talked about a lot. ACL-3 is a different flavor of BSL-3, so this is arthropod containment level 3. Means that many of the emerging infectious diseases, including Heartland virus, are spread by vectors, ticks, mosquitoes, fleas. And that is a really complex niche of biocontainment. And we can work with fleas, ticks, and mosquitoes at level 3 in our ACL-3 suites. We also have animal biosafety level 3, so that's an extra dimension where we can look at pathogenesis or vaccine or therapeutic efficacy in infected animals. That requires animal biosafety level 3. And we're just bringing online an agriculture version of ABSL-3 that offers even higher containment than ABSL-3. You will be happy to know that not anyone can just go and do this. So there are regulations that cover much of the work that we do. And for the most dangerous pathogens, there are federal regulations called the Federal Select Agent Regulations. And they're regulated by 2 agencies, CDC and USDA. And they dictate the design of the facilities, the operation of the facilities, the ways that one deals with a problem in those facilities. But also they come and inspect at least once every year, and they make provision for surprise inspections, and those surprise inspections do indeed happen. They also make sure that you know exactly how much of every vial of Eastern equine encephalitis that you have at any one moment. That, if you think about it, is really not a trivial exercise. It's something that's very important, but it's very hard. So putting this into scale, this is a -80 freezer, of which we'll have several. Everyone has several. There'll be a bunch of them in this building. This one has 6 shelves. On each shelf, there is 5 racks. On each of those racks, there'll be 3 trays. On each of those trays, I don't know, there might be 6 of these boxes. Each of these boxes can hold 96 vials. We need to know the location down to the location in the box of every one of the select agent samples. And there is no room for error. This has to be 100% accurate 100% of the time. And they will come and check that. And that is always the most nerve-wracking part of an inspection. But it is also the most high-risk area. So, I mean, I've not talked about vaccines and therapeutics really at all that we've done. I want to take an even wider berth real quick and talk about how it is important to have these capabilities in the country, the biocontainment capabilities, even when we think about unusual events that can happen during a pandemic. So we've been playing in several different areas. Firstly, during the Ebola pandemic when I was in San Antonio, epidemic, when I was in San Antonio, and then more recently, and when we think about SARS-CoV-2 and COVID-19. One of the areas that I think all of the labs spent time working on, and we were no different, was ultraviolet inactivation of pathogens. This is such an appealing approach that you have something that is passive that can kill as long as there isn't a shadow. So, for example, we did work with this company during the Ebola outbreaks, and we work with Clorox as well, who have a similar device. This is a robot that sprays ultraviolet light into a room. So the idea is they designed it for hospital rooms, so the robot just makes its way around the hospital room, much like a Roomba would clean the floor if you had a Roomba vacuum cleaner. And it shoots UV light all over the room. So what we did to test this, we had some Ebola on coupons, and we set up a dark room inside the BSL-4 and sprayed UV from this device onto the samples and determined how effective it was at killing. We did similar things with a number of different companies, not least Signify, which was Philips Healthcare, to look at inactivation of SARS-CoV-2 and other viruses as well. An area that I definitely didn't anticipate getting into was banknote cleaning. That it turns out that, I mean, I don't know that anyone really carries banknotes in this country anymore, but in other parts of the world, people carry banknotes. And they are cleaned. They're not thrown away when they're used. They're cleaned, and they are cleaned fairly frequently. And there is a real financial benefit for cleaning these devices, cleaning these notes, because it's expensive to keep printing the money. So the question from a number of central banks in a variety of different countries was, if these banknotes are contaminated with SARS-CoV-2, does our process kill the virus that's on those banknotes? So we had the company that makes the machines that clean the banknotes build one that we took into our containment lab, and we tested, and they also sent us money. And we tested those banknotes to make sure that the device killed the virus on the banknotes. The person that was doing this was very proud to say that he worked on a money laundering project. So then we worked with the TSA, and you know, often when you go to the airport, your bag will be swabbed with a little, sort of paper coupon, and sometimes your hands might be as well, like in this diagram. But it turns out that to save money, they don't throw those coupons away each time. They're reused. So the TSA and others wanted to know whether the process of putting it into the whatever it is that's detecting the explosives destroyed the virus before it went and maybe contaminated the next person. So we built one of the swab machines, or the company that makes them built one, and we took it into containment to ask if that machine killed virus. I put this on here because it shows that I'm really quite ignorant and unimaginative. So particularly during Ebola crisis in the mid-2010s, we did a lot of work on lateral flow assays. And there was all sorts of arguments for how these would revolutionize testing for Ebola virus and similar viruses. I couldn't see it. I couldn't see how there would be a value in lateral flow tests. And then when we get back to working with SARS-CoV-2 in the COVID pandemic, they are the thing that really saved us. Another area that we've spent a little bit of time playing with is with a group in North Carolina who are chemists. And also a company in Texas called Kraton. They make a paint that goes onto surfaces. And if there is a liquid component to anything that touches that paint, there'll be a very localized very steep drop in pH. So there is rapid acidification of the liquid that is deposited onto that surface. And they argue that this could be useful to inactivate virus. And indeed it was. We did work that allowed for EPA emergency approval of this particular paint, and this is an example of one use. Delta Airlines painted their check-in desks that allowed them to stay open during the pandemic. It was amazing how much interactions we had with the flying industry. I remember a call with Boeing, or a series of calls with Boeing, and they wanted to set up seats in the BSL-4, which is where we were doing it at the time, to mimic what was going on in an airplane. And they were going to send a device that they contracted with, much like this robot head, down the middle of those seats, and we were to spike the seats, put some virus on the seats, and they were going to spray the UV over the seats. It was remarkable. Okay, so vaccines. Really, I think Edward Jenner gets all the credit for starting vaccination. But people have been playing with it for a little bit before then. But the idea of vaccinating an individual to protect them from a subsequent exposure to a pathogen was around about 1796. And the individual was James Phipps, and it was captured in a very famous painting. But the vaccination for smallpox really, I think there's a strong argument that this is the most successful element of public health intervention ever. There was a lot of smallpox. I'll show you some numbers in a minute. But from the thousands of years that humans had been living in the presence of smallpox, vaccination was introduced 1952 in North America, mass vaccination. 1952 in North America, it became extinct in North America in 1952. 1953 in Europe, and then it's always the last mile that's the hardest. We have 1980 where it was declared free, but prior to that, the last case was in Somalia in 1977. So just bringing it back to me, down the road from where I grew up was the last known fatality with smallpox, which was 1978. And it was a medical photographer who was working with samples from smallpox research. And I think this highlights the need for biocontainment and working safely with pathogens. But the numbers are extraordinary when you think about smallpox. So you probably can't see it, but this is the number of cases reported worldwide. This number is 600,000, and we go from 1920 to 1980. So in the 1950s, there were 600,000 cases a year. And smallpox has a 30% case fatality rate. We're talking big numbers of people. Between 1900 and 1978, maybe 300 million people died. This was devastating. And it doesn't exist anymore. It exists in two places. CDC has smallpox virus, and the Russian equivalent to CDC at Vector has smallpox virus. They're the only two places in the world where it exists. So, viruses, I said, are small. They're not complicated. They don't have many genes. So if we think about the human genome, which we know is double-stranded DNA, this is, the unit here is kilobase, so this is 3.2 million times 10 to the 3 nucleotides. So 3.2 billion nucleotides is the size of the human genome. And they're of the order of 20,000 genes. Smallest virus, 2 kilobases, so 2,000 nucleotides, and maybe 2 to 3 genes. Rhabdoviruses, that's rabies virus, has an RNA genome, doesn't even bother having a DNA genome, carries 11 kilobases, so 11,000 nucleotides, and only 5 to 6 genes. I mean, remarkable how devastating something so simple could possibly be. Filoviruses, Ebola virus, 19 kilobases, 7, 8, 9 genes. SARS-CoV-2 is a coronavirus, 30 kilobases thereabouts, 7 to 14 genes. These are very simple organisms, and yet they cause such impact. They don't even have the ability to live on their own. I think a philosophical argument would be, is a virus alive? I think most virologists would say no, it isn't alive. It's not dead, but it's not alive. It can't exist unless it infects a cell to replicate itself. This is a problem. It should be easy to develop medical countermeasures against viruses, but it's not. It's a problem. They have so few proteins. There are so few targets that can be used by a vaccine or a drug. On top of that, especially when you think about drugs like to interfere with a process. But if the virus is using cellular processes, that makes it very hard to be a target for an antiviral drug. They also evolve exceptionally rapidly. They're a great tool to look at evolution. So I pulled out some numbers this morning as I was thinking about it, because we've had a long time interest in looking at viral evolution and how we may exploit it, but also what are the consequences of viral evolution. So if we think about Ebola virus as an example, every time the genome is replicated, so 19,000 nucleotides, there'll be 8 differences every time, at least maybe 8 differences. Very few particles are needed to infect a human and cause disease. But when the human dies, they might have 1 × 10 to the 13 viral particles. And that's not a static event. There have been multiple rounds of replication to that point. So there's an enormous capacity for in-host evolution. Doesn't mean it occurs. There is negative selection pushing back on the virus. But nonetheless, there is an enormous capacity for evolution. So in the last few minutes, I want to give you some examples, or an example of the sort of thing we work on. So vaccines, even though they've been around a long time, it's very hard to make a vaccine. It's the same for a therapeutic. So this number may not be exact, because I was not all that careful counting, but there are less than 20 viruses for which there are FDA-approved vaccines. And that might change very rapidly as we move forward. mRNA vaccines have the potential to really accelerate this sort of progress. But it's nonetheless very hard to make a vaccine. Likewise, for a therapeutic, it's probably harder. There are therapeutics for 10 viruses. Therapeutic antibodies, which have been around for several decades but have really come to fore in recent years, might change this dramatically. But when we think about emerging infectious disease, we're trying to think one step ahead. So it's not let's see what's coming down the pike and then we'll try and make a vaccine. We want to try and get ahead of the curve. So we want to make a pan-viral vaccine. So that would be a pan-coronavirus vaccine that works against all coronaviruses, including future coronaviruses. Same idea for universal influenza vaccines. The problem is it gets harder the broader you try and go with these vaccines. And on top of that, licensing might be tricky. The FDA might say, you can't have a vaccine for something that we don't know is going to come down the pike. And then you have viral therapeutics. It doesn't get any easier. It might be easier to license them. But as you get broader with your antiviral treatment, the efficacy, the effectiveness of that antiviral treatment tends to decrease. The best antivirals we have some elements of broad spectrum. But as you go broader, they become less effective. Acyclovir and valacyclovir are great for herpes simplex virus, but are less good for related herpes viruses. Remdesivir is okay for Ebola, works a bit for SARS-CoV-2 and COVID, but as you get broader, it becomes less effective. So to do this, we need to be collaborative. So science nowadays is much more collaborative than it was when I started. And I think this is particularly true, well, I mean, I'd like to think this is particularly true when we're thinking about trying to make these broad spectrum agents. We work with physicians, bioengineers, no need for me to go through this. But all realms of science, not just biological science, take part in our research. And I want to take you through, in the last few minutes, a collaboration we have just to touch on one area of the research. So this is a collaboration we have with a group in San Diego, and it's the group of Liangfang Zhang. And some of the work that I'm going to present here was funded by the Defense Threat Reduction Agency. The other bonus of collaborations is you get to go and visit these people. So I pulled out weather from San Diego, which is 75, it's like that every day as far as I can tell, and yet Colombia today is 0. So these are additional reasons, bonus reasons, why it is good to have collaborations. You know, he is desperate to come here. And I am not sure. I like it here, but not at 0 degrees. So Liang Fang is a bioengineer. He's not, and as far as I can tell, that means he's not really a biologist and he's not really an engineer. And within that subset, he's a nanoengineer, which seems to be a funding tool because anything nano is cool. So he developed and has patented what he's called cellular nanosponges. So these are small, tiny polymer particles that they have come up with a way of coating the polymer with a cell membrane. And that cell membrane can be natural or engineered, and I'll come on to that in a minute. But the point about using a cell membrane is that it's like a cell, just without it being a cell, and it can be manipulated. So like any other cell, that membrane can stick to bacteria, or it can stick to viruses. It can stick to elements of the immune system that might be dysregulated during disease and mitigate their impact. They're safe too. So these are in phase 1 trials. They're safely digested by phagocytic cells and they don't leave anything behind. So the idea is you have your starting cell, and we've done a fair amount with red blood cells and macrophage, and even mixtures of them. And that they are burst, leaving the cell membrane, and then a process that I imagine to be very complicated. They decorate the polymer particle with that cell membrane. So again, this is like a tiny cell, much smaller than a cell, but because it has all of the viral proteins and cellular proteins and lipids, but it doesn't have any of the cellular machinery. They can be generated in very high concentrations. So each cell yields thousands of these nanosponges. And this is the idea. And we started work early 2020. So early on in the pandemic, we were approached to work with these guys. So if we imagine this is a virus, and the virus has the goal of binding to this cell, entering and making more of itself, which will end up making us feel sick. The nanosponge, which is not quite drawn to scale, will bind to the virus particle. And in the process of binding to that virus particle, and maybe more than one nanosponge will bind to the viral particle, it will inactivate through mechanisms that we don't fully understand, but we have some ideas about. We'll bind to that virus particle and inactivate it. So this is science, but I won't spend too much time going through the assays that we use, but we use a fairly standard workhorse assay called the plaque reduction neutralization test, the PRINT assay. And what we published here was effective neutralization of the original variant of SARS-CoV-2. So this is from the epithelial cells. We have the percent neutralization up the y-axis and how much nanosponge we needed to achieve that amount of neutralization on the x-axis. And we typically report this as the neutralization concentration 50, or the inhibitory concentration of 50%. And we saw beautiful neutralization curves for both the macrophage and the epithelial cells. What was cool was it was variant independent. So we kept rolling through with new variants as they came through. And we would have predicted this, but it was still exciting to see. What I will say is that I work with small molecule chemists who have all sorts of very clever ideas about how antiviral agents should work. This is the simplest idea that you can imagine. You're taking a cell and removing all the bits of it that make it a cell except for the membrane, and you're trying to stick it to the virus particle. And yet none of the broad spectrum efforts from other areas have been anything like as effective as these guys. So we can tweak the system as well. So many viruses bind to a particular molecule in a very non-specific way on the outside of a cell called heparin or heparin sulfate. So in this paper with these data, we took cells and then we made nanosponges and we modified those nanosponges to have a higher concentration of this heparin sulfate on the outside. And sure enough, it was much more effective at neutralizing virus. So now if I break down the IC50 concentrations, we've gone from 800 micrograms per mil to a fraction of a microgram per mil. So really increasing potency. Can even go further. There are lots of different ways that we can do this, and this is the penultimate slide. We can engineer donor cells in a lot of different ways. So these data on the right show how residence time was improved. So really the half-life of the drug inside the host was increased with some minor modifications. We could also, especially if we knew what virus that we were targeting, we could overexpress viral receptors in the cell membranes that are used to make the nanosponges. And we've done a little bit of that, and that does indeed work very well. What we haven't spent a lot of time doing, but is coming down the pike, is to target specific tissues. So we can take a nanosponge and we can hit different tissues, which might reflect the viral tropism targeting those particular tissues. What we're really having fun doing now, and it's sort of iterative, but it is nonetheless trying to break the system, and we are failing to break the system, is to determine if it can work against more pathogens. And I'll tell you that we've tried it against a bunch of different viruses, and they've done bacteria and also cytokines, and it works against everything. We can improve things because we need to tweak the biology of these nanosponges that reflect the biology of the particular viruses. But I think we're now settled on a method that allows for very high potency against every virus that we have tested. So this really has the potential to be a broad spectrum, true broad spectrum antiviral agent. So lastly, the teams to acknowledge that I didn't have a photo of the team at TBRI, but I want to acknowledge the chair of my department, Jean Patterson, who really encouraged me to get into high and maximum containment work, and Ricardo Carrion, who is a colleague, and still works in that area. The team at BU, as I was leaving, they did a photo in a training room with everyone in a suit. We have the team at LSC, and I think everyone here is in the room. And then we have the team at LIDR as well. And I'm happy to take any questions.
How do you screen people to determine who's safe to work in the lab to make sure that a bad actor doesn't come in?
Some of them are in the room. I've got to be careful. So there are layers to it, and it reflects the risk associated with the work as well. But really, I think when it gets down to the root, we rely on trust and knowing the individuals who are going in. So it sounds trite, but putting team on this slide, I think is a valuable approach, that we all know each other and we all know what each other is doing. And if someone is behaving out of line with how they normally behave, or just out of line in general, we'll be able to identify that. But there are more official routes as well. So for select agents, that CDC USDA program has an FBI-administered background check that's associated with it. But there are other things too. But I would say it's down to knowing people and trust as the most valuable part.
Could there be any preventative aspect of this type of therapy, or is this really just a therapy once someone has already been infected?
Well, if you'd have asked me that 2 weeks ago, I'd have thought that it was post-exposure. But it turns out that they've tweaked it to have really decent half-life, sort of depot method, where depending on how it's delivered, it can hang around at what we hope to be therapeutic concentrations for an extended period of time. And if that's the case, I mean, if you think about, this is Department of Defense funding. So the idea would be that, and this is true for antibody therapeutics as well, you can predose a warfighter prior to going into a zone that you know to be of risk. But 6 months later or a year later, there's no obvious impact of that treatment. Henry.
Yeah, fantastic. So what's your next priority target?
All right, we can talk about flu. Yeah, I mean, actually, flu is where we're going. I think the breadth of it will be limited by how easy it is to manufacture at real scale. So I'm not completely convinced. Well, actually, I don't know. I shouldn't say that. I don't know that it can get to a scale where it could be of agricultural use. A 500-pound animal versus a human who isn't 500 pounds. But I'd like to try and play in that area as well. But it needs the ability to make more, lots of it.
Great talk. Thank you very much for this and for the work. I think you mentioned that the cells can be manufactured. So these don't actually have to come from the patient, be extracted from the patient, then manufactured into the nanosponges and put back into the patient?
No, not at all. So these are not primary patient-derived cells. And you probably can't do that. It's difficult to license and do that sort of thing at scale. So there are cell lines, immortalized cell lines that have more of an FDA blessing than other cell lines, and those are the ones that have been focused on so far. The red blood cells are slightly different.
So you had said that the Ebola virus could mutate 8 times with each division, and it multiplies to 10 to the 13th power by the time it kills the host, leading to a lot of evolution. How quickly can these nanosponges be made to adapt to some kind of mutation like that?
So I didn't talk about negative selection, right? So outside of negative selection, which is how many dead virus particles you get versus those that are viable and can replicate. Outside of that negative selection, every round of replication incurs 8 changes in that genome. It's much less than that because the virus can't tolerate that many changes. And Ebola doesn't evolve that quickly either. But one would imagine, or I would posit, that the steps that are needed for the virus to enter the cell are not that prone to mutation. Some of the steps involved in binding might be, but they're not that prone to mutations. I think something like flu, which is a bit more dramatic as it evolves, could be a different test, but we'll see.
I got a quick one. Can AI be used to predict mutations?
Well, it depends if you ask someone who does AI or not. I mean, to me, there are some great tools. So, for example, in that plaque reduction assay, I mean, we're discussing this in the lab right now, or at least I'm discussing this in the lab right now. We would go through hundreds or maybe, I don't know, Daniel, thousands of plates, particularly during the height of the pandemic. We were talking about renting a factory in China just to make plates for us to do these assays. And each each plate had to be counted. We had to look at the consequence of virus infection by hand, which really means looking and counting very manually. In our lab and in other labs as well, people have spent a lot of time trying to count these plaques, as they're called, using various technologies and generally in various degrees of failure. Sat down the other night with ChatGPT and taught it how to count plaques in an hour. So from that point of view, from tools, it can have value. I think on the bigger scale, we know so little about biology, whether it be in anything that goes on in this building in biomedical science. We know so little that I'm not sure there is enough information to feed AI for the very big questions. For the very specific questions, maybe we can train it to help. But big picture, I think we're a ways away. But I'm not trying to get funding in that area.
So I'm going to ask one last unusual question. So we've been talking bad about viruses. Has any of your work ever filtered into the positive effects of like gene therapy? So viruses as a delivery system, because there's a lot of things about, you talked about tropism, and particularly I'm interested in the nervous system. Has any of your work ever crossed into that domain at all?
Yeah, in a couple of different ways. So when I was a postdoc, we worked tangentially with using herpes simplex virus as an oncolytic agent, which certainly for glioblastoma, it has real utility in that area. More recently, I mentioned mRNA vaccines. I didn't talk about all of the vaccine modalities. There are several viruses, and it's not a coincidence that it's a virus. There are several viruses that can be manipulated and be very useful as vaccines. And we've done a lot in that area. I mean, certainly there are some very good vaccines that will be coming down the pike in that area. The licensed Ebola virus vaccine is a virus vector.
Fantastic. So lastly, we would like to thank you for your great work, for joining us here in Columbia, and for sharing your work in an accessible way and giving us your time today. So thank you so much.
Thanks, Dave. Thank you.
So one more reminder, no Discovery Series next month, but please join us in April. Thank you.